Start recording. Okay. I hope you don't hear the blinking of my messages coming in.
I picked up 7 folk from the other room.
Yeah, thanks for doing that, Allison. I was like, wait, usually we have more people by 5 minutes before seminar. So yeah, Tuba wasn't available, so she used my link from so I could manage the seminar. So hopefully with the email I just sent, that will reach people.
Yeah, I see we're up to 33, so.
Yeah, I think we're good. All right, so we can get started. Let's see, make sure. All right, so if you're not muted yet, please go ahead and mute your microphones. It's my pleasure today, welcome to seminar, to introduce our speaker, Dr. Darcy Telenko. Darcy has had a very interesting career. She started off in Southern Illinois, working with John Russin on SDS and tropical rots on soybeans. And then she went to North Carolina State University for her PhD, where she started off with Jack Bailey and then transferred to Turner Sutton because of her previous advisor's passing. And there she works on peanuts and white mold. And after that, she had postdocs and kind of extension positions at Virginia Tech, Florida, University of Florida, and Cornell, all on field crops, peanuts, agronomical crops, and at Cornell was 100% extension. And she can give you more details about that, but so very kind of interesting path there. And now she's an assistant professor at Purdue, where she has an extension and research position, very similar to Allison Robertson and Darren Mueller in our department. And she works on all field crops, but that primarily is going to be corn, beans, and wheat. So with that, I'm going to transfer it to Darcy. We actually overlapped a little bit in North Carolina State. That's how we first met. I was doing my postdoc and you were finishing your PhD in our little building there on the outskirts of the campus.
And I think that, I was gonna say, I think the building's gone, but thank you, Leonore.
Yes, welcome. And yes, we are looking forward to your talk.
Yep, well, I appreciate being here. I guess I just want to say I thank you for inviting me. I did hope I could come out to Iowa because I have yet to be out to Iowa since moving to here in Indiana, but maybe next time around. And just for grad students out there, I have had a diverse background in my career path. I actually followed my husband for a while, so that's why I postdoced and made it look like you could follow a naval pilot from Virginia to Florida. And then it was my turn, so that's where we ended up in New York, where I worked in 100% vegetables, and then finally landed us back here in Indiana doing field crop pathology, which is where I really wanted to be. But we've been here about 2.5 years now. I'm here at Purdue, and so I'm kind of just going to share a story of what we've been doing the last 2.5 years. So I'll share my screen here, and please feel free if you want to ask questions while I'm talking. We can do it that way, or wait to the end. Either works for me. Being an extension, I really miss that interaction, face-to-face interaction. So feel free if I go too fast, because I feel like I talk to myself now. But again, just an overview of what we've been learning the last 2 years. So when I started here at Purdue in 2018 in August, we had a new disease that started to impact corn. And so that's kind of what I'm going to give you today, is just a story of what we've learned and how, having this extension research interaction is very important, especially when we have some emerging threats that threaten our different crops. So just an overview of my program. We promote, we work on sustainable sound disease management in both soybeans and corn. Then I do a little bit of wheat, and then I use that information to generate extension efforts, that whether it be extension programming, extension writings, or outputs that way. So, when we look at the research and extension connection, the first thing is identifying and documenting the need. And so I'm going to talk about how tar spot developed into that, and then trying to develop research to answer those questions, and then the importance of sharing those results so then growers can implement what we learn. So again, tar spot of corn was the disease that landed in my lap in 2018. I didn't know anything about the Phylochorus species. But again, this is the disease that we see here in the corn canopy here in northern Indiana. And I'll get into more details as we move on. So in early August 2018, I started August 1st, and I received my first phone calls from the industry about this issue a few days later. As in an extension, we made, make the visits to the field to see what the problem is. And so this is that first field I went to in 2018. And you can see, the problem is here, the corn should be green. It's middle of August. We should have another month of it being nice, green and lush. And we got varieties that are going down and they're burning up. So as we continue to look at fields, we identified that we are seeing the tar spot, the Phylochorus species, but the emails and calls continue to spread in 2018. And so that was where we tried to identify what was going on, how widespread is it, and I'm going to share some of the information we collected. But again, as we get on to the crop canopy, you can see where we have this rapid blighting that's occurring. And if you didn't know the crop should be maturing at least another month, you might think it was beginning to dry down. But when we look closer at the leaves, we get this profuse amount of stromata that gets to be is produced on these leaves, and that's what's leading to that rapid blight that occurs within the canopy. I mean, we had growers that shared images here, just looking at a hybrid field day. They had a lush green crop one week, and they went back to those plots the next week, and a week later, it was just burned up with tar spot. So the growers and industry hadn't seen this disease. They didn't know what to look for. I suspect in that green plot in the upper corner, the stroma were there. It just hadn't turned to that blight stage of the disease. So when we tried to dig into the literature, what we come up with is that there's a tar spot complex. It's been around in Mexico and Latin America since the early 1900s. It can lead up to 50% yield loss in those susceptible hybrids and under those conducive conditions. And in the literature, they talk about the result of two fungi, the Phylochoromatus and the Monographelaomatus. And so initially, when we first found the tar spot pathogen here in the United States in 2015, we only had the one, we had the Phylochoromatus. And so the assumption was made, we only have one of the two pathogens. We really may not need to worry about this complex or causing significant yield losses. But again, in 2018, that's when we learned that we didn't need the secondary fungus to move in to really see those significant impacts. When we look at the Phylochorus species, there's about 994 that are named. They're all obligate biotrophs, which makes it a fun fungus to work on because we can't do much in the lab. Everything we are doing right now is relying on field work and data we collect during the season because we can't readily culture it. We've tried to inoculate in the greenhouse and we've had some successful attempts, but again, we only generate one or two stromata. We don't get the full blight and disease impact that can occur. So we're still working on some of that to see if there's a way we can better work with it to understand more of its biology. Most of the species are named after their host association. So there are Phylochorus species that have been on many of our weeds, but they're very species specific. So the corn, the Phylochoromatus is the only one that we know that infects corn. They have a typical normal narrow host range for each species. So the Phylochoromatus, of course, is the only species we know to infect the maize. It produces what we see here is these raised stromata on the foliage and husks. They're black in color. They can be very small pinhead sizes to very elongate. And they may or may not have that necrotic area that they talk about in the tar spot complex. But here in the United States, we have seen that necrotic area to develop. But again, we've never pulled the Monographela, and there's some work being done that has shown that other pathogens may be moving in, just using that area to colonize secondarily. What we know is they produce ascospores profusely when they're as the weather conditions are conducive. We also see some Conidia, which is the Lanacra species, and we're not sure what that Conidia does in the disease cycle. And so we don't think it's seed-borne a lot, and I think the movement may be more on just disease tissue. So this is where we found tar spot in 2015. These are just from samples that were submitted to our diagnostic labs. So we first found it here in Indiana and Illinois. And then we continued to see that year after year. But by the 2018, it had been confirmed both in Wisconsin, Michigan, Iowa, Florida as well. So Florida has a little hot spot that they see in their overwintering corn, and then Ohio. So in 2018, we are seeing a wider spread of the disease. And then this is where we had those significant yield impacts. So in 2018, where we got onto those fields that were significantly impacted, we estimated about 30 bushels per acre were being lost. When you get out into a field, 100% of those corn plants are infected, and we see 30 to 50% of that severity on that ear leaf that leads to that rapid blighting. And this is occurring around the R5, R6 stage. We got out into all our hybrids out and across those 4 different states where we had this significant impacts. And we saw that most of the hybrids were susceptible to infection at varying levels. And here in Indiana, we estimated about 3% loss. So 120 million lost to that year in 2018. So I had growers that think they've lost over 100 bushels per acre, especially between their irrigated and non-irrigated parts in their field. So we had a lot of concern here on the impact of this new disease and what it was going to do for the future. So what we knew and didn't know is that there's very limited information on this pathogen biology. We learned that hybrids may differ in their susceptibility. You know, fungicides could be used as a management tool, but there are questions on what's the best timing and what modes of action do we need to be looking at. And then there are questions on rotation and tillage. So here in Indiana, we've seen it every year since 2015. So we've had samples sent into the clinic, but it seems like we were building up this perfect storm for the 2018 epidemic. This disease really requires a lot of high moisture. And so that's what we saw a lot in 2018. From the literature, it likely overwinters in crop residue. Maybe there are other weeds or other plants, we don't know. And then in the literature that's talked about that those ascospores can be ejected up to 250 feet in that crop canopy. So if the inoculum's there and that residue surviving year to year, there's a high potential of getting it into the crop canopy once those conditions are conducive. So we started a collaborative effort with colleagues both in Illinois, Iowa, Wisconsin, and Michigan to try to look at the disease impact, pathogen biology, and management. And it's an interesting project that we started from ground zero on trying to figure out what we needed to do for this new disease. So a tar spot working group met in December 2018. We tried to meet monthly to continue coming up with ideas of what we wanted to work on. We coordinated research trials and tried to document the movement. And we worked alongside the corn IPM pipe and EDE maps, and I will show you some of those as we continue. We also started establishing fungicide efficacy trials, fungicide timing, and model development. So I just want to acknowledge that we have a large working group that's come together. This is the group in 2019. I think we'll be adding more in 2020 as I look at this, as the disease continues to spread. But I just want to acknowledge that some of this work is requiring a group effort to try to get a better hold of what's going on with this pathogen. So what we learned about the biology of Phylochora is that it is favored by cool, wet conditions. Generally 60 to 70 degrees Fahrenheit is what's said in the literature. And it has this long 7 hours of leaf wetness required at night. The spores are released predominantly at night. And again, they have that wide dispersal that can occur. And it probably is overwintering in the residue within the ascospores, which is very common for this genus. So the first thing we did in spring of 2019 is we tried to collect residue across the various states to really see, can we see that this pathogen's overwintering? We may not be able to culture it, but can we get ascospores out and can they germinate? So this is the results of that collection. We had samples from Illinois, Wisconsin, Michigan, Indiana, looking at the ascospores from a sample and then the germination from that. And it was really interesting results. So some of the samples had over 4 million ascospores being released in a milliliter of water. But there was an inverse relationship. The more ascospores that we were seeing released, reduced germination. So even in some sites where we may have less ascospores, 30,000 or whatever, we were still seeing about 12 to 20% of those ascospores germinating with 4 hours and up to 24 hours. So this did prove that yes, the Phylochora is overwintering here in the Midwest. It is surviving our conditions. And that's going to be providing that initial inoculum for years to come. So we saw about 33,000 to 4 million ascospores per milliliter of water being released. So that's a lot of initial inoculum that could be available for the next season. And 1% to 25% are viable, at least based on their germination. So again, once we had these hot spots, we now have this inoculum source that is readily available to cause disease in future seasons. But looking at the disease, again, we only have the Phylochoromatus that seems to be impacting corn here in the Midwest. So we're really just looking at a disease cycle with the Phylochora. It's overwintering and residue. Again, ascosaurs are being released when we have those conducive conditions, wind and rain dispersal. And then it takes about 14 to 21 days, we're estimating, before we start seeing these raised stromata being formed on that leaf tissue. And sometimes the stromata may or may not be enclosed by the necrotic halo, which is called a fisheye. So sometimes we see the fisheye, and we've seen it every year here in Indiana, but again, it's a very minor part of the disease. Mostly we're seeing just a profuse amount of the stromata forming on the green tissue in the plant. And then after that initial infection, we see the repeating cycle of infection. We really don't know the time frame, but it definitely seems in my mind, those secondary cycles seem much faster than that first initial infection period that occurs. There are questions on, is there a leaf age that's required before it can infect? Is there a time of year? So we still, the more we get into this, I think the more questions we have, just because we don't know much about the biology of this fungus. But the one thing, being an extension was, looking at COVID and wanting answers right away, is our growers were seeing this significant impact on their fields in 2018, and they really wanted answers the winter of 2019. As we turned into 2019, what do I need to do this season? So we tried to address some of those issues in the short term. So again, in 2018, I arrived and I made an effort not only to look at the clinic samples, but tried to get out into Indiana and see where we could find tar spot. So here's just a map that we were using to track tar spot conformation in Indiana. And when I got to looking in the fields, we had a couple hot spots, the northern part of the state, where we have lake effect weather from Lake Michigan. And then we had a hot pocket around that central part of the state, Shelby and Rush. But when we get out there, the thing is, growers had never seen this disease. So we had to first initiate, how do you identify it? What are we looking for? And there's a range of symptoms that we can see. There's a range of amount of stromata that can form on the tissue. So this is just an example of some of the different leaves that we collected. Sometimes you may have large stromata and 4 to 5% infection, whereas other times there may be millions of little ones on a leaf, completely colonizing every area of green leaf tissue that's available. And I also show here that we could go out into the field after it had been harvested and look at the residue and see if we could find the stromata in the tissue. So as we turn around the corner and started in 2019, everybody was really nervous about tar spot, and so they actually got out and scouted a lot. And so we had a lot of initial reports that talked about, they were thinking they found tar spot, but there are things that look like tar spot. So insect frass was one of those that we get a lot of calls about early in the season as they're out trying to scout. And so we talked about, with insect frass, you just wet your finger, you scrape at it, it's going to come off. With the tar spot stromata, it's embedded in the tissue, you scratch at it, it remains in the leaf. And then if you think you have it, you can send us a picture or image and we'll take a sample and try to confirm it. We also, Physoderma brown spot with its purple-black lesions may also mimic tar spot. Generally, the disease doesn't appear till later in the season. So maybe some of our old, mature southern or common rust pustules can turn black with age. And so they may, later in the season, we had a lot of southern rust this year. So there were false positives for thinking they had tar spot. But the other thing I think is where we might have missed, where it was slowly spreading around Indiana after 2015, is that it does look a lot like our saprophytic fungi growth as the corn's beginning to break down, and especially since it's moving in later in the season. So I still could go out in the field this fall and collect some of that tissue, and the stromata look a little different from the saprophytic fungi that grow on the tissue. So it's one thing, they can still go out and see if they can find in the residue if they're worried that they had it in their field. But the other thing we're working on is when do we first see the disease appear? And so in 2019, we tried to get out in some of those hot fields that they were planted back to corn again and tried to see if we could scout and find the tar spot stromata and when was it initiating its disease cycle in the field. So this is just an example of what it looks like to find one of those first few tar spot stromata. It's a needle in the haystack. We were out in that field about 30 minutes. I was ready to quit and move on to the next field. And that's where I hit this first lesion. Again, we only found one that time. If you look at it, it has this raised structure. You scratch at it, it's embedded in the tissue where that stromata is growing. And so after making that first find, I sent my students back the following week and said, okay, you guys roll out with me. We only found one, see what you can find and how much is it spreading week to week. And this is the image of the pictures that they found once they went back. And so we go from having very little to find, it's a needle in the haystack, that first infection, but a week later with the conducive conditions, we can have hundreds of them forming in the lower canopy. And so there's a question of, how fast does this disease develop within that crop canopy, and when do we need to implement those management tools? And so those are some of the things we're trying to look at as we try to answer some of these early questions on using fungicides and other things. And so this season, out in our fields, we set up a camera just to show you the change and how fast the disease progresses. And so I apologize, this is my first attempt at trying to put together a time-lapse video, but I thought I'd try to show you what we captured this season on, a 20-day period from when we got stromata just forming on the green tissue. You really don't see it unless you're looking for it. And then how rapid those leaves will be blighted. So if I can play this, let's see here. So you can see here, I got a leaf that has the stromata formed on it. There's a little chlorosis surrounding them. But as over time, you begin to see more black spots, the leaf begins to turn blighted, and but it takes a little bit, but you can see you get larger chlorosis, necrotic area. And then eventually, once we get enough stromata there, that's when we see the significant leaf blight that occurs, this early rapid burning up within the crop canopy. So you can see it's turning here, and then we'll lose that leaf as it drops down. And as you see this video progress, you can see the other leaves that may be infected, and slowly over time, you see where the upper canopy above the video camera drops down into the frame. You can also see some of these leaves in the back where they're blighted already from tar spot. So it's just kind of an interesting, within 20 days, when the crop should still remain a little bit more green at the end of the season, we do see this rapid blighting. And that's where we're thinking the yield loss comes in, is that the crop is just not getting to its full yield potential on that yield. So you can see we have this rapid blighting that occurs. So just as an example of some of the tools we're trying to show growers on how fast it can change. And that's some of the calls that I've received. As you know, the corn was looking great. We went to Florida for a week. We came back and the whole field was burned up. And so that was a lot of the calls we were getting in 2018 and 2019. Again, in those hot fields, we have profuse stromata forming in all the tissues. And it can infect the husks of the corn as well. So any green tissue out there can be infected by the Phylochora species. So again, this is some of the hard hit fields in 2019 where you can see that blighting that's occurring in that crop canopy. So as a multi-state group, we began to track the distribution. So this is just a map showing how we monitored it. So those initial infections would have been sent into the clinics. And then as we were out trying to scout in 2019 and 2020, we expanded that area to how widespread it has moved. And we'll have the first reports coming out of Pennsylvania and Canada this year. So again, from the initial infection in 2015, it's spread now to 10 states and Canada. So we continue to see this movement of this disease through the United States. So when we're talking to my growers, I'm trying to determine the risk of tar spot in Indiana, what parts of the state are most at risk, and the questions of what's influencing this annual epidemic, and can we use some of this information to help monitor the disease and help predict where it's going to happen in the future. So my team made an effort to do a statewide survey here in Indiana, supported by our Indiana Corn Board. So we were able to get out and scout at the end of the season of 2019 and 2020. And it's the hard part with this is the disease moves in late. So we really have to wait until we know the disease is active in order to get in the field. So in 2019, we were trying to catch fields before they were harvested just to really document. So we didn't get into all 92 counties. But in 2020, this year, we were able to get into those fields. And what we did is we tried to catch 2 fields per county. We rated not only whether we found tar spot, but took that field incidence. How easy was it to find? Did we have 100% incidence in all those plants? Or was it 1%? And then on those leaves, what was the severity level? Because I think that tells us the disease may be present in many of our counties, but it may not reach those significant levels that causes yield impacts. And so we try to track this disease annually and in season with the EDD maps, so we can provide extension updates of when we know the disease is active, it's in your county, maybe you need to go out and look at your fields to see if it's there, to try to guide our growers into making a fungicide decision. So this is just an example of, the distribution of tar spot in Indiana. And so the first 3 years, of course, it's just samples that were sent in where growers noticed a problem in their fields. But we added up to 16 counties. By 2017, we had confirmed tar spot. In 2018, we were able to find it in 25 new counties. So we got up to 41 counties in Indiana. And then 25 last year, we added again. And then 2020, we've added 12 new counties. But I can say in 14 counties with our survey, we were unable to find it. So we still have that southern edge of Indiana where we may not be readily able to find tar spot, but it does continue to move. And I think it's going to continue to spread throughout the Midwest. But as we got in the counties, so when we look at the EDD maps, when we find tar spot, we turn a county red. Now, the positive notation tells us, yes, the inoculum's there, but it really doesn't give us any information on the severity of the disease. And that can range. We can turn a county red or yellow, even if we find just one stroma on an ear leaf or a leaf in a field. And so that information is good to tell us that, yes, the disease is present, it's just laying low, but it's really those fields that where we have the significant spread of the stromata where we see the yield losses. So where we have leaves that are 25% or more of severity, those are the fields that, we'll see the significant yield losses. So we try to, map this out and try to look at the range of severity, looking at both the ear leaf severity and then also the field incidence. And we came up with an index for Indiana, both for 2019 and 2018 or 2020. So this is the index that we developed in 2019. So it kind of shows that we have a real hot spot, Port or LaPorte, the northern part of the state where we have the lake effect weather, where we have long periods of moisture, long dews in the morning. Those are the areas where we have the highest amount of disease. Those are the areas that are most at risk that may, need to be actively scouting to make a fungicide decision. As for, the central and southern Indiana, really the disease is there, but it's not really going to cause enough problems to lead to any yield losses. It's just good for the growers to know that, keep your eye on it in case the weather conditions change. We could see that rapid shift from, a few stromata to numerous numbers. So when we got into 65 counties, about 89% of the fields had tar spot when we checked it in 2019. For 2020, the map changes a little bit. So we got into all the counties. We had about 50 negative fields, so about 75% of those fields were positive. So again, 78 of our counties had tar spot. We added 12 new ones, but there are still 14 counties where we couldn't find tar spot in those fields that we checked. That's not saying it's probably not there, but again, it's a lower risk for our growers. But again, as you can see, the hot spot remains up in northern Indiana, but we are starting to pick up some other areas that may be of concern to keep an eye on for the future. But so the other thing I wanted to show you is just breaking this index down. So if we look at just field severity, so the darker orange, 100% field incidence down to less than 1%. So we were only able to find one plant or so with tar spot. You can see that generally it's northern Indiana where we have, we can find tar spot in every plant. It just may be varying levels of severity. But in 2020, you see, we're starting to see a shift down the river there that we might start see some areas in Vermilion, Vigo, and Sullivan counties that might start picking up more disease in the future. And then looking at the leaf severity here in 2019 and 2020, you can also see that, 2019 and 2018 were really bad years here in Indiana for the disease in those northern counties. I would say this year, the disease didn't move until later. And I'm not sure we'll see the yield impacts that we saw from previous seasons. Again, it's because that severity, we still have green leaf tissue available. And so I think our yield impacts this year will be much less than they were in 2019. And if I take and look at the weather conditions, we had a lot of drought conditions here in 2020. So this is looking at the end of July and the end of August here in Indiana. And you can see, as compared to 2019, we had some significant drought areas in most of the state in July, and then in the northern part of the state in August. And I think not having those high moisture conditions really limits how fast this disease can move in the crop canopy. So again, if you look at all those maps from the 2 years, again, we're seeing some shifting of areas that may be hotter pockets that we need to keep a closer eye on, but it does show us that, you know, northern Indiana is the part of the state that we're really most concerned about for seeing any yield loss impacts for this disease. So some of the things for the survey we've learned is, you know, this tar spot has continued to spread in Indiana. So we had 7 counties from our peak DL samples in 2015. We're up to 78 of our 92 counties in 2020. We do see a range of severity. So I think we're at low risk central and southern Indiana for tar spot to really lead to any yield impacts. But we have a high risk here in northern Indiana. But there are some areas where we're maybe starting to build up more inoculum. So we're going to keep a close eye on some of those in the future. But the biggest thing is weather conditions are going to continue to play a big significant role and influence this annual risk. Here in Indiana, tar spots here, it's going to be here year after year. So we just have to learn how to manage it and what conditions maybe make it more conducive for disease to develop versus other years. So some of those questions we look at is, you know, how do we best manage that tar spot? So we went out into a lot of our hybrids in 2018 and tried to look at their susceptibility to the disease. And so we compiled data from Illinois, Indiana, Michigan, and Wisconsin, looking at our performance trials. And we grouped them according to relative maturity. So we have a 92 to 106 day maturity group and 107 to 114 day. And what we were seeing in general is we saw about 0.48 bushel loss per 1% increase of that ear leaf being infected by tar spot in that early maturing group. And then in the later maturing group, about 0.8 bushel loss per 1% increase in tar spot. So every 10% we see of that leaf being consumed by the tar spot, we almost see about 8 bushels lost in that later maturing group. This correlated to some of the yield data that we were hearing from the farms across the region. And I just want to show an example. This is an example of 2 hybrids, same maturity group, planted next to each other. So you can see there is some variability in the hybrid susceptibility. Both are infected, but you can see that one is just the infection, the amount of inoculum, the disease infection occurring in that crop canopy is much higher than the other. I suspect in 2 or 3 weeks later that the one on the right would be as blighted as the one on the left. But again, there is good news that some of our hybrids may be able to hold it back a little bit and allow them to get to full maturity before the disease takes over. And this is just another example of a field in 2019. And you can see the differences in our different hybrids that the grower had planted. So you can see there was a difference between the maturity groups, but also if you look in the 97-day maturity group, there are some hybrids in there that show a little more susceptibility than others. So it is important we tried to encourage our industry to try to get out and rate some of those hybrids for susceptibility because that could help manage the disease in the future. So in 2018, we did document that we have a range of hybrid susceptibility to the reaction. We saw about 40% to 50% tar spot severity in the ear leaf by many of those, which led to about 16 to 38 bushels per acre loss. And then our growers, that reported, we estimated 20 to 60 bushel yield loss from that season. So turning to 2019, we started to coordinate fungicide efficacy, timing, and integrated disease management trials across the region. And so I'm going to share some of those results that we have accumulated in the last couple of years. So first, I just want to say I got lucky here in Indiana that we had a field station up at Pinney that had significant tar spot developed. So we have a field station that we don't have to inoculate. We have natural inoculum that we can try to monitor and track this disease. And so what we tried to do is evaluate the percent of the stromata that occurred within the crop canopy, and also then the symptomology, the blighting, the chlorosis that occurred. And we rated both on the ear leaf and the ear leaf plus and minus 2 to try to get an idea of how the disease was moving up in the crop canopy. So in 2019, we first found tar spot around that station on the 13th of July. We began to monitor it. It remained relatively low till about R3, R4. And then we saw almost this exponential increase around R5 of the disease within the crop canopy. So this 2-week change really scared us because it's a rapid change from only having 1 to 2% of disease to 30 to 40% of that leaf being consumed by the stromata. Looking in 2020, the disease shifted a little bit. We really didn't have active disease until a little later in the season. And then we tried to rate further out into the season as well to see how far, if we could continue to see the disease moving in the crop canopy even after we reached R6. So again, the disease this year was not quite as exponential, but again, we have this rapid change where we go from 2 to 5% of stromata to 20 to 25% of that stromata in the lower and the middle canopy. In addition, when we look at the symptomology, the necrosis follows, right behind. Again, the canopy stays relatively green, and then all of a sudden it changes as well as we see this continued infection within the canopy. And both years it looked about the same. It's just a little delayed here in 2020. So this is where I'm thinking we may not see that significant yield impact this year as we saw in previous seasons. But this is the rapid change that occurs in our plots within almost 2 weeks, is we have plots that have there's stromata there, but we really didn't see the rapid blighting. We go back 2 weeks later, and that's our non-treated plots. So this is what has really scared our growers throughout the northern part of the state. So in our trials, we just have a standard randomized complete block design. We identified a nice hybrid that has good resistance to all the other diseases. So we really could look at just tar spot. We do apply irrigation in some of the trials just to try to promote the disease, except when we're trying to model it and look at timing for fungicides. We use a Lee sprayer for applying our fungicides at 15 gallons to acre. And then we rate the disease all season long, looking at the leaf tissue. And then at the end of the season, rating percent green and lodging, and of course, yield metrics. So we have a number of the products that we use in corn evaluated in these trials. I'm just sharing the list of some of the major products we have out there. Most of them are mixed modes of action. We did throw in tilt, which is just one. Looking at the common fungicides that are used in corn, will these work at managing tar spot? And so here's the first trial in 2019. Again, similar graphs is what I've shown. Disease appeared on the 13th of July. We started with our standard application timing window of a VTR1 application. That's generally what we go out to control gray leaf spot or northern corn leaf blight. We wanted to see, will this window work for our growers? Will it provide the control that we're looking for? And so we went out at August 8th and put the fungicide sprays out. Here's our non-treated disease curve, and then the fungicide curves following. So as you can see, most of the products, they worked really well at shifting that disease curve down. They protected the crop for those 2 to 3 weeks where fungicides are active. But you can also see at the end, once those products run out of steam, the disease begins to take off in the crop canopy. And so this is going to be important when we're looking at timing, is do we need to shift this application depending on the season? Are we losing yield at the end of this disease curve where it's moving up in the crop canopy because we haven't quite reached R6, we haven't reached black layer? Is there still yield potential that we need to protect here? So we ended up in 2019 having more questions, but the good news is most of those fungicides did significantly reduce tar spot as compared to the untreated control. Similar in 2020, I just compiled this data yesterday. We again went out with the VTR1 application. Again, our disease curve is not quite as exponential, but the fungicides relatively managed to keep that disease down through the R5. But we're starting to see a little separation here, and I think that's the longevity of some of these fungicides, is how well did they knock the disease back. And so we, from this data, we might be able to separate out, the different areas where some of the fungicides may be more effective than others. Looking at the area under disease progress curve, again in 2019, all the fungicides performed similarly. And this is what we saw in most of those trials across the region. If we break it apart, based on the canopy. So we rated ear leaf +2, ear leaf -2. Again, in 2019, that VTR1 application, all the fungicides were able to hold off the disease and reduce it significantly as compared to the untreated. When we look at 2020, we are starting to see some products separate out. Some of the new products, the Veltima, Revitec out of BASF, seems to hold on a little bit longer. So we're starting to see maybe we might have 3 groups of products, some that may provide a little bit of control, but maybe not as effective as some of the other products. So this is just data we just pulled out of the field. And hopefully I don't have yield data to show you today on the 2020, but hopefully by winter time, we'll have compiled some information with multi-state data to really delve into some of these products may be more effective than others. But again, we did this, looking at the same thing on the ear leaf and the ear leaf plus and minus. Again, some of these products really are reducing that disease curve as compared to others. But my question in 2020 is, the disease moved in later. Do we see that significant impact on yield as we saw in previous years? So the other thing we like to rate is looking at the leaf necrosis and the stain green potential of the fungicides. And so the goal is we want to keep that crop canopy as green as long as possible to get to that black layer. So this is 2 different ways to look at it. So here's the necrosis. So you again can see that most of our fungicides do reduce that necrosis development. They're protecting that crop canopy, which corresponds to keeping the crop canopy green through the end of the season. So again, we're consistently seeing that many of these fungicides are providing consistent results in reducing the disease and keeping that crop green in order to get it to its full yield potential. Looking at yield, so I only have 2019 data, currently starting to harvest our trials here today. But for the 2019 trial, the first trial in Indiana, we really didn't see a significantly difference in yield, but if you look at it, there is a range of 3 to 21 bushels per acre with a fungicide application. This is one of the trials we had out. I went and pulled other trials to try to see if we are seeing any significant yield bumps with our fungicide. So those trials where the disease bumped up a little bit more, we were over 30%. So this one's at 39% in that non-treated control. We do see significant yield impacts. So this one ranged from 17 to 20 bushels an acre. So in general, if we had significant amount of disease, the fungicide, well-timed fungicide at that VTR1 did provide about 17 to 20 bushels per acre where the disease occurred. When we looked across some of all our trials here in Indiana, 7 of those 9 trials, we did see significant yield bump at that 17 to 20 bushels. And the severity occurring on the ear leaf was about 35 to 40%. So those trials where we did not see the statistics significant yield bump, the tar spot only got to 4 to 24% of the ear leaf. So again, there may, in trying to determine the return on investment for our growers, it's really those areas we know we can protect the yield if the disease gets out of hand. But again, on the lower end of the spectrum, we may not need to be putting out a fungicide for controlling tar spot. The other thing we noticed at Pinney was that there is an increased lodging risk. As the tar spot is shutting it down early, we're starving that stalk, the corn is starving it to try to finish that crop. And we did see, especially in 2019, that a lot of around the plots, we had significant lodging risk. What I told my growers though, is we need to cut open those stems. So here you can see the stem is clean. It's probably mostly due to tar spot occurring within those plots, but I was out in field calls. So here's just another example of field in 2019. Significant lodging had occurred. They have a ton of tar spot occurring in the field. But then if you get down and look at that stalk, we have every other stalk pathogen moving in. So it is important, to understand that may not just be one disease affecting a field or another. We need to look at the multiple pathogens and make sure we understand what's really affecting the issues that are occurring in a field by field basis. So in summary of our fungicide trials, all the fungicides we evaluated in 2019 and 2020 are effective in reducing tar spot. We are starting to see maybe some differences where we can separate out some of those products. We'll be combining trial data from other locations to see if there's any consistent trends. And then we're going to continue to evaluate products and timing as new things come onto the market. So in our second bigger project, we looked at timing, and I'm almost out of time, aren't I? 5 minutes, okay. So I'll just cover timing. So we tried to look at, is there a better application timing? So we used TripAPro, applied at these various timings. And after 2019, we learned that we had a lot of growers that wanted to go late. They wanted to start applying fungicides at R3, R4. Even this year, I've had growers saying tar spots in my field. We're at R5, should we go? So we put those timings into our trials to try to look at that answer in 2020. And so here's the 2019, and you can see those early applications. The fungicides are running out of steam before the disease really kicks up, whereas that BTR2 application really helped, was the best timing for that year. And again, if we look at those AUDPCs, we can significantly reduce tar spot with that BT or R2 timing. Now in 2020, when we added, and we saw that same kind of yield effect, that BTR2 is where we're seeing that significant yield boost with the fungicide application. And this was consistent across the multi-state trials. So we combined them all. And again, that VT to R2 application timing really is working best as looking at the trials across the states. We are working on an app in collaboration with Damon Smith in Wisconsin on trying to look at, can we model and predict when the disease is going to be rampant in a field? And so we've just tried to modify and work on that model. It hasn't worked as well in Indiana. We added a rain factor and that increased our precision in 2019. Again, the model never clicked for us in 2020. So we're still trying to figure out what's the difference between Wisconsin and Indiana. But I just wanted to share the one thing that I discovered, this hot off the press data from the season before I run out of time. And so this year we went out with more timings of application. Again, that early vegetative V8, V10 stage really doesn't do anything to the disease curve. Our VTR2 look like they're the best timing, again, for this season of reducing disease. And then when we go later, that R3, R4, and R5, really the R4 and R5 really didn't impact the disease curve for this season. Our double hit, the V8 followed by the VT, works relatively well as well, but really we're not seeing a greater separation from just the one application at the R2 or VT stage. And then our tar spotter never triggered. So we still had a negative, or we didn't have that application out for Indiana this year. So with that, I think I'm going to open up to questions before, since I ran out of time, Leonore. So I will stop my share. So I get too excited and ramble on, too many slides. Go ahead, Mark.
Hi, Darcy.
Hi.
Nice talk, really enjoyed it. Maybe I missed it, but it seemed like there's a big gap between Mexico and the Midwest, and there's a lot of corn in between. So where did this come from?
That is the big unknown question. So the initial, if you look at the initial report out of Indiana, they were hypothesizing that a hurricane moved it up, but we get hurricanes every year. So what shifted, and maybe it's just we got a different pathotype that survives in our climate, changed enough, so there's that aspect. And then there's the other one of, I think moving residue could have been a problem, right? And I think that's another thing we might need to consider. Did it come up in a, in seed somehow from one of the areas? Not on the seed, but I'm thinking just, we've gotten samples in the clinic of seed and I've been able to find tar spot lesions and leaf material that was left in those samples. So there's two thoughts. We don't really know where and how it got here and how it established. So that's one of the big questions. And I think we'll have more questions knowing where it jumped to Pennsylvania. So it jumped way into the middle. If you looked at that map, Pennsylvania this year. And so they're wondering, how did it skip those corn acres in the middle?
Thank you.
And so it is one I wonder, should we be monitoring for it? Should we have been monitoring for it a long time ago to make sure we didn't get it introduced here? I don't know. Roger?
Yeah. Hi.
Hi.
I had a question on host range because a few years back, I had my Carl Forster ornamental grass in the front yard heavily infected. I didn't know what it was, and I brought it into Lena at the clinic, and she had a colleague she sent it to out east, I think. And I don't remember who that was, but I think she's on the seminar. But anyway, I wonder if this was just specific to corn or just grasses, or because they identified this and the other reed grass, this ornamental grasses looked just like the corn did. It was just completely dead.
Right. So there are 900 plus species identified, and mostly are on the grass, weed species, and other hosts. So we have 900 phylochora species, and we're still trying to determine what really separated them out. And most of the information is just based on that host pathogen, not looking molecularly, just looking at that interaction microscopically. I think trying to think who's working on, but I think they're trying to dig apart or separate out those different phylochora species. But the Matus, well, as far as we know, Matus only goes to corn, and we really don't see it spread to the other weeds. But we're still colleagues are still trying to.
She asked me if our grass was near a cornfield, which it is down the street from one, but I don't know if that was ever resolved. That's why I wonder if it was just going between the grasses, or it's actually you're saying it's a different species.
Yeah, so there's a phylochora of Graminis, and there's a whole bunch of other ones that we've pulled at some of those weed species and looked at them. But again, I'm not on that aspect of that, but other colleagues are looking at to try to tease apart how related are they.
Okay.
That sample went to the ARS mycologist.
Okay. Yeah, I think they're working on trying to tease more apart of what's going on with the phylochora genus. And did it come back Matus, or it came back something else?
I haven't heard, now that I think about it. I should share an email.
No, it's been kind of exciting to work on a new pathogen, other than it's really hard to work with. And so we can't answer some of these basic biology questions other than what we can try to collect in the field year to year. So we're trying to answer those low hanging fruits. As fungicides work, will timing work until we get a hybrid with good resistance, and trying to develop some of those.
Well, this variety or species is virulent on a very popular ornamental grass in the Midwest.
Okay, and that could, that might provide some
you see this particular species planted all over the place, in people's yards, gardens, everything.
Could I ask another question? Could I ask another question, Darcy?
Sure.
Were you done, Roger? Okay. It's early in the game here. I mean, and epidemiological knowledge and practical knowledge, but has anybody approached doing economics on the, trying to figure out on an average, maybe even 5 years, is it going to make economic sense to put on a fungicide for this?
So I would say I have a student working on, that's going to be part of her work, is she's going to try to pull in the economics. We pulled in some of the our economics faculty here to try to help her look at that, to see, protecting 20 bushels, that pays for the fungicide. Some of the new products are more expensive, and in the promotion of hitting it twice may not get the return on the investment. I have growers that feel like they got ahead this year and they hit it twice. And so that's a lot more than we're used to seeing, especially with the prices but that's the conversation we're having. When we talk about fungicide timing and making that decision, is the disease really there and active? And is it really that return, is that investment timing, is that investment worth it? And then that's the timing aspect. If we can best time it to only put one fungicide out, you may want to hold it to R2. If nothing else is pushing the disease, other diseases aren't pushing things along, you may want to hold on to it so we have that later season protection. So that's something that's different with this, is we want that later season protection. But I don't want them spraying 2 to 3 times, but I know our chemical guys do. So
well, I assume that eventually this might be linked to some forecast weather information to sort of make the decision as to when to pull the trigger, right?
Right, and so that's what we're working on with, Damon Smith has pulled in his white mold app to try to bring that structure in, to try to pull in the weather data and try to fill that in. But again, we're basing off a lot of this biology, is based off what was going on in Mexico, and we're seeing different things than what they've reported. So we're starting there, but I feel it's more active here in Indiana when it's hotter. It's just, I think there's a bigger moisture factor than temperature. So, and those fields that are wet or we have irrigation, those are the ones that are going to get hit year after year.
Thank you.
Yeah, I've got a question.
Okay.
What do you think are the challenges or limitations to growing this pathogen in like greenhouse or growth chamber settings?
I think it's trying to figure out how to best get it to grow. So it's an obligate. No one's been able to, and I question what we've tried. And so it's just having the time to try to tease it apart. Are there other ways we can try to get it to really grow? So I still think one of the things we're hoping to start this winter, and it is just trying to build programs, but it seems like there needs to be a fluctuation in the stomata opening and closing for it to move in and out. And so we're trying to build a chamber in the greenhouse that gets it wet at night and then dries during the day to more closely mimic what's going in the field, which is not generally what we're set up to do. We usually keep the relative humidity at one level. And so I think we need a fluctuation that we're seeing in the field. That's my thoughts on where we need to try to tease it apart to try to get some greenhouse or lab work going. And so that's kind of where we're stuck right now until we figure out how the best way to get it to grow. I mean, we can get it in the greenhouse, but we get 1 or 2 stromata and that's it. So that really doesn't tell us anything on trying to, tease it apart.
So is it like in the field, is it just you get a few initial stromata, then you think they reinfect? And that's
right.
not
So I personally think, we have that initial infection. It takes some kind of environmental conditions or plant growth stage. And then that secondary cycle seems to just go crazy and much faster once it gets going. So I wonder if there's a difference in the spores that are being released, the overwintering versus the secondary spores. But again, we're just getting started. And I only have a couple, still getting the lab off the ground here. So.
Yeah.
But yeah, I think if we could figure that out or what that secondary cycle, but it seems like you go week to week and all of a sudden it changes from, 1 or 2 lesions to like hundreds. And so I showed that picture where you couldn't hardly find it. And the next week there's, something triggered everything to infect. So.
Thanks.
So Darcy, maybe I'm wondering if there are other people with other species have done any studies where they're looking at maturity level, so plant developmental stage, to see if that sudden increase has something to do with the physiological maturity of the plant, or if you had enough inoculum in the right conditions, especially in the greenhouse. You probably don't want to grow corn up to like mature corn
Right.
before you get an infection. So that might be some interesting studies to do, just kind of inoculate these corn plants at different stages and seeing if that's part of the reason it picks up, or if it's just a question of the inoculum buildup.
Right. And I think it's a combination. So we did try to get some corn infected. And so I would say since 2018, we went and planted seeds in the cornfields that were hot, and we were able to get infection right on the cotyledon. So when it's moving in that crop canopy, we could get infection every part of the plant. Cotyledons were infected. But I still think that initial primary infection, I think we have to get a mature leaf, or it's not, maybe it's infected and it just is not appearing. Maybe there's a longer latent period than we know. But then once it gets ramped up and it says, hey, the conditions are right, we can just move in it moves faster. I don't know. I say we have a lot. I have more questions than I have answers, but we're trying to get at least some fungicide answers out there so the growers can do something to try to minimize the impacts when we have bad years. So.
All right, does anybody have any final questions for Darcy?
I would say, we did have I didn't show this, but we do have I did have some hybrids that showed a little moderate resistance, and using that eliminates the need for the fungicide in some of our trials. It didn't give us the additional benefit. So if we can get good resistance out there, then we may be able to walk away from using fungicides. But, we're still a ways away from, because none of our breeders were looking at this disease. So.
Great. All right, so we're a bit out of time, so I'm gonna put up. If anybody wants to stay on and talk to Darcy, if you have a few extra minutes,
I'm here, yep. I'm not
you
going
log
anywhere.
off, that would be great. So
Thanks for doing this, Darcy. Appreciate it.
Oh, no, I appreciate it. Next time I hope to come to Iowa.
Absolutely. You're welcome.
yes.
I promise I won't bring any more tar spot, but Darren found it across most of the state, so.
You, we won't be able to say it was you.
Right.
All right. Well, thanks again. We'll give you an applause, kind of silent. Just wave,
Thanks,
everybody. Thanks so much for doing this. It was a great talk. So we'll, yeah,
Thank you.
let everybody log off. Thanks.
Thank you.
Oh,